Looks like we're ready to go. Okay, well, we're going to get started here today. We got Randy Oliver ready to go. I don't think he's the guy that needs much introduction. We had him yesterday, but we all know who he is. He's going to give us some insight on his research on pollen supplements, probiotics, and then he'll have some time for some question answering. Now, if you do have any questions going throughout this presentation today, a lot of your screens, if you look down on the bottom, there's a question and answer section. Click on that, you can type it in, and we will see what we can do to make sure we get those questions answered for everybody. Randy, it's up to you.
Well, good morning, everybody. Well, I've been cranking data and making graphs since 5 o'clock. You guys are going to get some of the fresh off the press information here. Let me share my screen. There we go. Slideshow. Come on, from the beginning. Okay, what I'm going to do here is, hey, if one of you guys, if could put your picture on, I can watch to make sure you guys, there's nothing crazy happening. Okay, I'm going to show you. Yeah, there you go. I'm going to cover the first 3 of these this morning, and then I'll do these second 2 this afternoon. So I mentioned this very briefly yesterday about this thermal treatment. I thought I'd go in a bit deeper here today. And again, yesterday I mentioned this particular slide showing how the brood nest temperature for Apis mellifera is actually warmer than the ideal reproduction temperature for Varroa destructor. And the thermal tolerance, where the mites get seriously stressed at lower temperatures, then the adult bees have a very good short-term temperature tolerance. As far as the mites in the brood, to kill them, the issue is you're right at about the temperature where you start to kill drones. For one of the researchers who published on this said, "Oh, well, beekeeper, we can't go that high because the beekeepers couldn't stand to lose the drone brood." But in actuality, many beekeepers cut out or killed the drone brood intentionally to suppress Varroa reproduction. So that may be a trade-off would be willing to make. Let me tell you first, for the commercial beekeepers, I can't see thermal treatment being cost effective just because of the labor involved. But for the hobby beekeeper who is opposed to using any kind of, putting any kind of substance into the beehive, they have a lot of interest out there for this thermal treatment. So my image data looked really, really good at alcohol wash counts, showing thermal treatment just almost mite counts going down to nothing. So I was very interested in that, just because this looked very, very promising. So when a beekeeper sent me a thermal device to test out, I started doing a deep dive into it and wound up with a few of them and doing a lot of research on it. And this device did work. The bees do come out. And then I confirmed that we got a very high temperature inside the hive and that it 6 degrees for the 2.5 hours that it was supposed to do. They put the thermistor underneath the sealed brood in the upper box to that indeed the sealed brood was killed. And this is a big point. Speaking with the manufacturer of this particular device, is I tested it on doubles. The claim was that it works well on doubles, and with more conversation, it works really well on singles, but it may not get temperature up there to the 2 doubles. Although I in this case, I did confirm the temperature. We did hold this at least 106 degrees Fahrenheit for the 2.5 hours up in the upper brood without a problem. Then I did dissect out the mites. I think I showed this one yesterday too, where these mites in drone brood, patch of drone brood, the mites were, most of them were still quite active and very much alive after treatment. And it's here on the ratio of the live mites to the dead mites. And in all cases, but this one small drone brood, the number of live mites far outweighed the number of dead mites in these treatments, both in the drone brood and in the worker brood. So it appeared that, again, confirming some previous research done in Germany, that particular temperature regime of 160 degrees Fahrenheit for 2.5 hours does not kill most of the mites in the brood. And as you can see, this is reflected by my own alcohol wash count for the 5 colonies that I tested, where indeed the infestation rate of the adult bees goes down a little bit after 24 hours, surprisingly 24 hours actually, even more by day 15. But then the remaining mites, which some manufacturers claim that it has sterilized the mites. If the mites were indeed sterilized in the brood, you would not be able to see a quick recovery of the mite population in the hive. So this is not to say that thermal treatment cannot work. And this is what I came up with, what's called the thermal death time curves. And this is the way that these treatment devices need to be, or should be, effectively be designed, understanding these thermal death time curves. And this should actually be a 3-dimensional graph with humidity put in. Also, right now I just have the temperature and the time, and the 3rd dimension should be humidity on here to find out where this sweet spot area is that we can kill the mites without doing too much harm to the bees in the hive. So it's not the end of the story. Thermal has potential, but the devices on the market could need to start looking at this a bit more scientifically to hone them in. The next thing I did was a test of the probiotics. And I just saw an advertisement for a human probiotic on your sales ads a little bit ago. And these have great sales pitches. And when I go to the conferences where the manufacturers of these are, and they see me coming towards the booth, they smile and just shake their head because they already know what my question is going to be. Have you got supported data for this yet? And the point is anybody who's going to sell something to beekeepers, if they have hard data, they're going to have that hard data plastered all over their website, all over their booth. If there's no hard data benefit to these treatments, that means that if they're not showing that hard data, that means that hard data does not exist, that it's just a sales pitch without supportive evidence. What I looked for on this was supportive field evidence, because I've had a lot of requests by beekeepers, a lot of questions of whether these direct fed microbials or DFMs are of benefit to the bees. So I tested the 2 main ones that are on the market, the Super DFM by Strong Microbials and the DFM. And I've spoken at length with the manufacturers of this one right here. And then I tested them against a control of powdered sugar base for both of these treatments. I had a visiting scientist take the bags of these and go off in another room and put them into jars marked A, B, or C. So I am completely blinded as to the treatment here, and I still am blinded. And until I work up all my data and come to my conclusion, I won't unblind myself, which is the best way to run any kind of trial, to blind yourself. And I ran this trial from July through November, feeding these treatments once a month. Tested in 2 yards. One was a sunny yard, and one was a shady yard at slightly high elevation. And it's always good to replicate any kind of trial at least 2 yards. Then we went out, and at the beginning of the trial, we went out in the early morning when it's still cool and there's some cluster. We graded all the hives for start and end strengths. Well, it wasn't that cool, so the clusters were fairly loose. So, but this kind of grading, this cluster grading, which we use for almond grading, it's still good for comparison from hive to hive. It may not be accurate for the absolute value for how many bees are on the frame, but it certainly is good for a comparative value to compare one hive to the next. And this is my grandson, Eric. This is my son, Eric. This is my grandson, Ryder. And He has turned out to be a really good field technician for recording data and checking every hive number and making sure that every treatment is applied properly and all the data is recorded. In all of our field trials, we always have a technician just in charge of the clipboard, recording every step that we do out in the field. Then I welded together a modern hand truck with a cradle from motor and a $600 digital scale right here. So we can just roll this right up to a hive, shoot underneath it, pull down on this handle, and bingo, we get a very accurate weight to within a tenth of a pound. We fed the probiotics once a month, sprinkling them over the top bars of the hive. This is one of our first feedings as we were putting the second brood chamber on these hives. All the hives are numbered and color coded per treatment. And then what I did is at the end of the trial, we compared the colony strengths and the weight gain. So these are starting and ending strengths. And this is for treatment group A, treatment group B, treatment group C. So one is control, and two of them are the two different treatments. Notice the colonies did not grow much. We had this really hot weather I'm going to talk about later. So that really suppress colony growth in the late summer. The other thing is that we did the final grading when it was much cooler, so the clusters were tighter. So it makes it look like they grew less than they actually did, but that doesn't make a difference. We're looking at the comparative values here. So there's the difference between the starting, the blue column, and number of frames of bees here, and the red column, which would be the number of frames of bees. What we're looking for is a difference in the amount of space between the red and the blue for each of the groups. And as you can see, in the shady yard, there was no apparent difference between the treatment groups. So what we would say is there is no apparent benefit to gaining colony strength. For the sunny yard, we can look again, see this variation, and you have a few colonies that grew pretty well, but they occur in various groups. But the overall difference, again, I still haven't run the full statistics, but the initial statistics show there's no statistical benefit over the natural variation. So that's for colony strength. So this evidence does not support that there's a benefit to colony strength gain. Then we'll go to the weight gain. Weight gain was more obvious because we did do supplemental feeding with sugar syrup, and the amount of weight gain per treatment group was almost identical. And that's for the shady yard. Let's look at the sunny yard. And again, essentially identical weight gain. So once again, my field data does not support the claim that there was a benefit to weight gain of the colonies. Now, that still does not mean that there could not some benefit from feeding probiotics. In the bee's gut, there's a cluster of bacteria that live right up at the mandibles and in the hypopharyngeal glands. A very, very few of them crop. Some live in the midgut, especially some yeast in the midgut, but most of the bacteria are in the hindgut of the bee. And there's a core microbiome of 7 different species of bacteria, although none of those species are found in those probiotics. So the manufacturer doesn't expect us to see the probiotic actually established in the bee's gut. But that still doesn't mean that it can't have some benefit to help the gut bacteria. So I'm working with Dr. Kirk Anderson at the USDA lab, and I just sent him $5,000 to help pay for the next-gen sequencing of these bee samples, and I've sent him a whole bunch of samples to from which frozen on dry ice, and very expensive, like $500 a pop, shipping them overnight to get them while they're still frozen. So one of the questions, do any of the applied bacterial strains establish in the bee guts? We don't expect to see it, but we will see. More relevant here is, does the feeding of either probiotic affect the community structure of the gut? Now, this is a possibility, and we're going to see that. The other one is the claim that it helps these to resist nosema or resist European foul brood. So we can screen for both of those pathogens and see whether there's a difference in those colonies due to treatment. And then the last question was, many beekeepers are buying these probiotics under the assumption that if they feed their colony antibiotics, which kill bacteria, that by feeding a probiotic, it will help the gut microbiome to reestablish itself. So I was going to do this in the first part of the trial. I decided I'm going to split the trial into two parts. So once we got the final grades for the weight and strength, we then started a independent trial with the same colonies which have been fed these probiotics during the summer. And we divided each of the treatment groups into 3 subgroups. One subgroup got 3 consecutive treatments of tylosin. The other subgroup got 3 consecutive treatments of oxytetracycline, and the other one got treatments of powdered sugar. So now in each of those probiotic groups, we have treated them with antibiotics. And we took samples after the end of a few days after the 3rd antibiotic treatment, when we would expect to see the gut microbiomes wiped out. And then we then took samples again, and then we fed a probiotic treatment again after the antibiotic treatment, whether it would help the gut microbiome to establish. And we took B samples again at 7 days afterwards and at 21 days after to see whether any of the probiotics helped to reestablish the gut microbiome. So that work is in progress or soon to start. All the samples are frozen at the Tucson lab. Okay, another separate trial was this test of the pollen subs. We beekeepers very much depend upon pollen subs. It's interesting, depending on the group. I spoke to Pennsylvania a couple of days ago, and they have all kinds of stuff blooming late in the season. And then up in New York, I was talking to them, and they have this huge pollen and nectar flows late in the season. Well, California does not. We're more probably like you guys in Utah, where we have these very dry summers, and there's nothing for the bees to eat late in the season. And for 25 years, I moved all my colonies to the state to better pasture during the summer to build them up for almond pollination. And then we learned how to use pollen sub here in California. And in 2013, I ran a comparative trial of the pollen subs. And since there's 2 new subs on the market, I decided to run one again. So the sub that we've been using for some years is one specifically made for California beekeepers by Man Lake. It's the bulk soft product that comes in a bulk form in a 50-pound box. When we get out to the yard, we just dump it out and then chop it up to whatever size pad we want with spades. And it's soft enough to squish between the frames, between the 2 brood chambers. So we feed it right in the middle of the cluster. And we, in our operation, we typically feed a 2.5, 3.5 pound patty at a time. The problem with trying to feed the preformed 1 pound patties is you can't put three of these often, so that it's within the cluster. The bees won't touch any patty that is hanging outside the cluster. So that's why the bulk form. But the other ones all came in patty form, so we tested against that. So this is the bulk soft right here. And 2 of these, and then this one here, the homebrew. There was a group of commercial beekeepers down in the Bakersfield area of California, almond pollinators. And they've been worked with a Mexican nutritionist and came up with a formulation they've tested, and they're pretty proud of it. I think it works really well. And curious as to how their homebrew formulation compared to the formulations on the market. So they were major funders for this to help me cover some of the costs of it. Now their homebrew formulation also contains natural pollen. There's only 2 formulations that contain natural pollen, the homebrew, and then this one by Global, was a Canadian company, has a plant in Montana, and they have produced pollen sub for a long time. And I asked them to send their top tier product. So they sent the one that has natural pollen in it. So just these 2 are the only ones that contain natural pollen. Then Dadant has a new one out called AP23, which I wanted to test. Mega B is a product developed by Dr. Gordon Wardell when he worked for USDA, and his son now sells this product. Ultra B is the Man Lake flagship product, very similar to the bulk soft, but different. It is a different formulation, and we see the bees react to it differently in our hives. And the last one is this new spirulina algae product by Healthy Bee. And I communicate a lot with the manufacturer of this, and was involved in some early testing. This is the only one that has a high amount of essential oils added, especially thyme oil. And my question was whether that high amount of thyme oil may have had deleterious effects upon the colony. And I'll let you guys see later on. And then to compare these, we need to control the sugar in these patties. So I made up individual patties weighed out, matched to match the amount of sugar, average amount of sugar in these patties. This was the sugar negative control with no protein, but just the equal amount of sugar to make sure that the feeding of the sugar wasn't an issue. As you can imagine, it can be a little bit confusing to feed 8 different types of patties out to 144 hives in 3 different yards. So in all, we fed 18 hives for each patty type, divided by yards, so replicated in 3 yards, which again, very important because we got different results in the 3 different yards. So what we did is before we would go out on feeding days, and we fed quite a few times, and also fed sugar syrup, I got these shoe tubs right here and marked them with colored tape and color coded all of the patties. So we didn't name the patties when we were feeding at all. We just went by the color code, and the number on the front of the hives all got a piece of colored tape. We would then lay out all the patties on top of the, make sure there were no screw ups at all, that everything worked out right. And then we would start cracking the hives and putting the patties in. We chose the 3 test yards for yards that historically we knew that the bees would just nosedive if they weren't given pollen subs. So we took our 3 worst yards for summering bees, and we ran the trial in those 3 yards with enough hives to have enough competition that they could not get enough natural pollen. We got at 2 of the yards, we had a little surprise pollen flow for about 2 weeks, and the bees responded immediately. But other than that, the main nutrition by far was the pollen patties. We fed with syrup from August through February, early February, before we went to almonds here. And what we do is the patties that contain natural pollen were somewhat more attractive to the bees for consumption. Now, that consumption doesn't mean benefit, it just means they like to eat them. So we fed ad libitum, and we would go out every few days and check. And when the patties of most attractive ones were almost consumed completely, at that point we would feed the entire yard, and then we remove any uneaten patties and record how much was eaten. So we can now have a figure for the total patty consumption for each different patty type. And again, same as with the probiotic trial, we just could not get colonies to grow. It was so frustrating. And I was wondering what it was. And I talked about temperature yesterday. So I found a local weather station that was close to the 2 lower elevation yards, compared the 2020 high temperatures, daily high temperature, to the 2019 temperature when the colonies grew in those yards with pollen sub, and also the humidity low for 2020 for that day, and for 2019. And as you can see, this is our feeding period, and we're talking late September, October. Now this is when normally when we get these colonies as it cools off to just build up their brood nests big time. 2019, when they did build, how much lower these temperatures were down here. We got temperatures over 100 degrees during this time, and at 100 degrees, those colonies shut down. They're just, once you feed them, they just will not grow brood. So they have brood all the time, a small amount, but they would not. The colonies did not expand. Humidity is also a big factor. If you look at 2019, look how high the humidity came up a few times right here, which allows for that expansion of the brood nest. 2020, the humidities were much lower. So what I'm seeing is we're at the 2020 temperature humidities, we're looking to be around the thermal tolerance for Italian honeybees. If we had Carnioles, they would have probably been even more negatively affected. So, frustrating thing for us as climate changes and it gets hotter. So, this is over my lifetime. I showed one of these graphs yesterday. I showed the winter one. Here's the summer one. You can see how much more steeply the temperatures are increasing over my lifetime here in California as compared to the winter temperatures here. Okay, so then I look for the correlation between the amount of sugar or the amount of protein or the amount of lipid. I had all the patties analyzed, and there's no correlation with lipid for the change in colony strength. Negative correlation with sugar in the patties. Very strong correlation with the amount of protein in the patty. So protein was very important. And we also found natural pollen in the patty was not critical. One of the patties, which has no natural pollen, was in the highest performing group there. And in the analysis, looking at the different patties, the amount of protein ranged from 22% in the Ultra B down to about 14% in AP23. This is not the way the patties rank by performance. So it's not just about protein. And I will return to that. That's what I'm working this morning. It's how the protein is put together. So all the formulations based on the literature, which is all based upon one study done in 1953, suggests, and that study was by a guy named Grote, de Grote, whoops, back early in the days, and here's the recommendation for the amount of each essential amino acid, this black column, in a pollen sub. And you can see for all the patties tested, they were at or above the recommended amount. So based upon the literature, they all go, wow, yeah, our patties look just fine. I am questioning that. I'll return to that. The other question was, well, I didn't say, when we started the trial, assigned the treatments, we went to each yard, graded all the hives for starting strength, and then did a randomized block design where the first block of patties was assigned to the strongest hives in the yard and randomly assigned for each patty type. Then the next 8 strongest were randomly assigned 8 treatments down the line. So all patties were evenly distributed across starting strengths of the hives. So the question was, well, do the stronger hives grow better than the weaker hives? And this shows you colony strength for the starting strength versus the change in colony strength. No, actually the weaker hives grew a little bit better, stronger hives. And we started out with about a 8 frame average in these hives, okay, giving them room to grow. Okay, so at the November midpoint, this is the colony performance of the median change in cluster size. You see most of them went down, but that was mainly an artifact of the temperature. So don't worry about that. Just look at the comparing them. And so the top 3 that came in were the global, the both soft, and the homebrew. Both soft having no natural pollen, these 2 having natural pollen, and then no natural pollen. And what we saw was with the healthy bee with a very high amount of thyme oil in it, that appeared to be because it was right there around with the sugar control colonies as far as performance. So this is at the November midpoint before they went into winter. They then took a very brief break for about 3 weeks or a month. I got to check the figure, where they stopped eating. We stopped feeding them, and then in mid-December we started feeding them again. Then we just finished grading them February 1st. They took them to almonds. So this is after 6 months of feeding. And here is what I did here is I added up the total frames of bees that went to almonds. So this is the no excuse graph. This includes the dead outs, everything. This is a preliminary analysis. I don't go by this completely yet. But for the 18 hives, I added up for the grading how many frames of bees there were in those 18 hives going to almonds. This is your total number of frames right here. That's what we get paid for. We get paid on frame strength contracts. So this is our applied grading, which means how much dollars the beekeeper, us, would get by these. And we lost money big time on these two right here. Again, see the healthy bee in control, essentially the same high amount of die out of these two colonies right here, and very good performance, especially on here, even better than with our bulk soft that we have used for many. But this is very interesting to me, and this homebrew formulation, yeah. And I asked the beekeepers to make this if I could share their formula. And they hesitated at first, and they talked among themselves. They decided, yep, they're willing to share. So I will be publishing their basic recipe for our use here. Now, here's the sad part. Back around 1950, we were first starting to look at amino acid composition and essential amino acids in animal diets for all kinds of livestock, chickens, pigs, cattle. Nowadays, you can buy a specific formulation for your cat, for your chickens, that at every stage of growth, there is a formulation with a specific essential amino acid balance ratio formulated for them. This is constant research. You can go on the internet and read the updates all the time. The state of the art for the honeybee industry was published in 1953. This paper here is 80 pages of text and graphs prior to starting the many pages of citations. Lived in this paper for a number of years. The guy was an absolutely brilliant and meticulous scientist, but he was very cautious throughout, saying, this is preliminary research. Don't run too far with this. This is just some lab test. And yeah, he pointed out the deficiencies in this research, and nobody has yet, since 1953, followed up on this or analyzed it. So I'm going to be publishing a full analysis of his findings and what we need to have researched. I'm hoping to collaborate with Vince over at USDA, and maybe he will pick this up and move us forward to try to catch up with the rest of the livestock industries. Using what I've learned from this paper, here, I just put these slides in this morning. This is everything we know right now is based upon this graph. These are feeding studies where he took groups of adult bees, newly emerged adult bees, and fed them in a artificial diet of nothing but sugar and essential amino acids, not essential ones, all the amino acids, and made each diet deficient to see what kind of ratio should be in it. You're looking at the sum total of what all of our feeding recommendations are based on these days, right here. This is it, guys. Really very tedious back then, especially in the 50s for them to do this. I have software to grow, but this is really crude stuff right here. So what I worked for weeks on analyzing my data, trying to figure out why some of these patties perform better than others, until I had a brainstorm at 4:30 one morning, and I said, what if I added up the percent deficiencies of the essential amino acids relative to, I'm not going to tell you what it is right now because I'll be publishing this, and correlate that against the ending total frame strength of the bees. And look at this correlation right here. When you get an R-squared value of 0.84, that means that my prediction accounts for 84% of the variation. That's an incredible thing that makes a scientist really feeling pretty secure about this finding. That these are the top two performers are up here, and I could predict it that they would perform better than all the rest, looking at the amino acid deficiencies. So I'll show you something in just a minute. So this is very exciting, and I have already shared with the manufacturers that I will be helping them out, trying to come up with better pollen subs for us. Now, Dr. Vasigliano, I've sent him off samples, also samples at the beginning and the end of the trial. So he's going to see whether, first, he's already analyzed all the head and thorax weights of those bees, and they correlate exactly with the performance of the colonies, which means that they better utilize the protein in them. So he's going to see if it changes difference in the gut microbiomes, that the thyme oil had caused any problems. And he's looking also into other deep dives in molecular biology in the genetic up and down regulation. He likes to go deep in this stuff. So we may so I'm going to publish the practical paper. He's going to publish, we'll co-author a scientific paper here. Okay, so let me stop this share, and let me go to other share here. Oops, hang on, let me change one thing. Okay, back to Zoom, and back to a share screen. Okay, somebody tell me, do you see a new screen with a sheet on it? Go ahead and unmute yourself, guys.
Yeah, yep.
Okay, great. Okay, so here's how this spreadsheet works. Let me delete that. Okay, here are the Groth's recommended ratios of the different amino acids. Essential amino acids would be in there. Now let's compare them to what's in royal jelly. So I'm just going to put a 1 in here. The green columns compare the royal jelly to his suggested ratios. You notice it's pretty damn close, right? Okay, how about a mixture of known nutritious pollens for honeybees? Look at that, almost exact comparison. You guys getting this here? How close the bees are adapted to these, this is like brassica and almond pollens, nutritious pollens right here. Now, what you can do, let's just say we put in corn, 1 part of corn gluten meal. Wow, we got some major deficiencies right here. So we'll put in some soy, let's do 1-in-1 soy protein isolate and corn gluten meal. Well, it works out well here, but we're deficient right here. So we'll add, now do a 3-part formulation. Let's say 2 parts of brewer's yeast. Oh, that seemed to help a bit. Oh, maybe we should add some skim milk powder. Let's put a 1/2 part of skim milk powder in. This is a calculator that I put together to help beekeepers and manufacturers try to balance out their formulations until they match that of the Groth, royal jelly, or nutritious pollens. So this is going to be we'll be sharing with our industry, and you can put it in parts like this. You can put in 50% this and 30% this, and then we'll put in 40% of this. Oops. And it says, oh, you exceeded 100%. You can't do that. Okay. So you can use this any way you want to try. Oh, wow. Wow, that formulation actually doesn't look too bad, whatever, right there. Anyway, this is for everybody to play with, and I will be making this freely available soon. Okay, let's get back to stop my share so I can see you guys. And we've got time for questions now. Okay, I don't have time to read the chats. One of you guys wants to read the chats to me, you can go ahead. Yeah.
There's 4 questions posted, or 5 questions now posted in the question and answer.
I'm opening them up right now. Randy, yesterday I asked about the flea collar type varroa treatment. He said is by Bayer, not using the US. And why? The reason why is that in the mites that are resistant to, well, first they did it with Kumafos, and I don't know why they didn't follow up on that. And then the next one was with flumethrin. And the resistance mechanisms for the pyrethroids in the US and Europe are different. It's different genetics for resistance. So in Europe, flumethrin, the mites do not have resistance to flumethrin, but they do have resistance to fluvalinate. Here in the US, they tend to have resistance to both fluvalinate and flumethrin because it's a different mutation in the mites. In Canada, flumethrin also, the mites have a different population there and more are not resistant to flumethrin. So this is, that's the answer why, that they just didn't see a market for it. Here we go. Janet Brisson, huh? No, nose of it would not be considered a probiotic. Okay. Okay, next question. Do you add 2 to 2.5 pounds of pollen sub all at once? Oh no, we do 2.5 for every feeding. People think that putting a 1 pound pollen pad in a hive is anything other than an appetizer or fooling themselves. That's an appetizer for a hive. A strong hive that is hungry, they'll eat that 2.5 pounds of pollen sub in days. Bam, they just blast through it. More generally, we feed it again after 7 to 10 days, but bees can go through a lot of pollen sub very fast. A 1 pound patty is a teaser. How soon do you place your bees in almonds? Do you start to stimulate feed before you place your bees in almonds? They won't do much in our climate at my elevation, typically until around Christmas time, depending on the temperature. And that's when we started feeding this year. When we see them start to consume patties, then we'll feed them. January, the first week of January, if the weather's warm, we get an alder pollen flow. And if they have alder pollen coming in, there's no sense at all feeding pollen sub. The alder pollen is all they need. So we had a short alder pollen flow. So we stopped feeding for 2 weeks, and then the rain came in, so we continued feeding. And then after we place them in almonds, if the trees have not started, we hit them down there in the orchards with a 2.5 pound patty of pollen sub, so that they keep building before the trees come to bloom. 24 methylene cholesterol. I ran a trial last year. It's on my website of the results, which I would have bet money that was going to be the answer. No, it's not. Very interesting thing is of the lipid content of these pollen subs is the one of the best performing ones, the global, has the lowest lipid content by far of any of them. So that means that it would not have a lot of sterols in it. So that's another surprising finding. I've heard you talk about feeding dry sub in the fall, but potentially being harmful. Yeah, and that's just potential. That one very small trial, but not go far with that. That needs to be replicated. I was actually corresponding with Ian Stepler this morning about him feeding a dry pollen sub in the springtime, making a suggestion to him that the behavior, his rationale, I was making suggestions about bee biology and his rationale, that it's a good thing, but I questioned his logic on it. So we're that. What do you recommend feeding with pollen sub and from what form? I don't make any recommendations. When in our own operation, it's more cost effective to dry feed it. It takes less labor, but you get much better colony to script buildup and evenness by feeding it in patty form, but it does take more labor. What about the effect of freezing mites versus heat treatment? Well, if you freeze the mites when the bees are in the hive, you're going to freeze the bees also. So that would be not worthwhile. If you take the combs out, yeah, then you heat treatment or freezing or formic acid. Once the combs are out of the hive, then it's much easier to do a heat treatment. What about dry pollen sub? I did not study that here. I did make some recommendations to Ian as far as some other subs that he might dry feed other than the cornmeal he was talking about. Fires affect my business. Other than increasing demand for our nukes this spring, for those beekeepers who lost bees, we had to go in and emergency evacuate 4 yards of bees ahead of the fires. And we actually, animal control has to escort us in through the police line to do that. We couldn't get to one, and the fire got right up to the asphalt road and did not jump it. So we did not lose any bees. Okay. Should we conduct a study of the impact of hot tea with honey? guys, I start chemotherapy in a week and a half for this tumor in my thumb. It's getting in my on my tongue. It is getting painful right now to talk. I'm hoping the next few days I'm going to record all the Zoom presentations from this point on and not be able to do them live. Are all natural pollens equally beneficial in terms of amino acids? No. There are some that are much better, like the Rosaceae, and the have very good essential ratios of essential amino acids. Others do not. I think I got through all the questions. Any others, or you guys see anything else?
There is another one here, Randy. What about the, well, there's two more. What's the homebrew formulation pollen treatment commercially available?
No, but I will publish the basic recipe.
Okay, and then what about the 24-methyl cholesterol?
24.
I think he
Oh, no, go ahead. It's on my website. I thought for sure that was going to do it. I had a custom patty made by Man Lake using borage oil as a 24-methylene cholesterol source, and found out that my findings confirmed what was published back in the late 60s from the Tucson lab, that bees are able to convert sterols. So even without 24-methylene cholesterol, they can convert other sterols to 24-methylene cholesterol. So based upon my research, it's not necessary to have 24-methylene cholesterol in a pollen sub.
Okay, well, that's all the questions that we saw. So if anybody has any others.